Welcome to this introduction to rare disorder diagnosis with Geneyx Analysis. This introductory video provides a high-level demonstration of Geneyx Analysis's rare disorder workflow for analyzing a single exome sample. Geneyx Analysis offers powerful, knowledge-driven analysis capabilities and variant interpretation, leveraging the GeneCart Suite knowledge base. This includes phenotype-driven prioritization and scoring, phenotyping association evidence, automatic evidence-driven reports. In this demonstration, we will show how Geneyx Analysis can be used to identify a causal mutation in a rare disease. We will use a published case involving a 4-year-old girl in a consanguineous family with no gastrointestinal history that developed persistent secretory diarrhea at 18 days of age and has been completely dependent on intravenous nutrition. She had minor dysmorphic features but no obvious developmental delay. After creating a case in Geneyx Analysis and uploading a VCF, we will start the analysis process. The main analysis screen is designed to provide the analysts with immediate access to all the information required about the case. On the top right, we have access to summary case information, including subject information, clinical information, as well as information about the sample. On the top left, you can find a set of tabs dedicated to analyzing the case. The first tab, called Fast Track, provides an ACMG and phenotype-driven workflow for rapid identification of potential causal variants. In the majority of cases with a clear candidate mutation, you would expect to find the causal variant in the first few lines of this tab. Fast Track applies 3 filters on the VCF: ACMG classification, VUS or above only; quality filter, removing dubious variant calls; and ClinVar filter, removing any benign or likely benign entries. The variants are prioritized by default based on the strength of the gene-to-phenotype relationship. The next 3 tabs display variants based on the basic genetic models: recessive homozygous, where we will focus on homozygote variants; recessive compound hetero, where we will be looking at genes with 2 heterozygous variants; and dominant hetero, where we will assume a single heterozygote mutation is sufficient to cause the disorder. Mitochondrial variants, for which specific annotations are available, have their own tab. Finally, following ACMG guidelines, we provide a tab for incidental findings based on the ACMG list of genes. In this demonstration, we will be using the Fast Track tab to analyze the case. On the left-hand side of the variant table, we provide a control area called Filters and Tools. This includes quick access to the list of predefined filters, including the ability to remove or add filters as needed. Importantly, your account admin can easily create and modify various filters based on your needs. Column filters may be added by clicking on the filter icon next to the column title. You can also see and modify the list of phenotypes keywords entered for the analysis. In this specific case, we've selected diarrhea, parenteral nutrition, and FTT. You'll notice that when I enter keywords into the system, it will provide a visual indication of the strength or uniqueness of the keywords. In this case, you can see that diarrhea is probably associated with thousands of genes, and therefore it receives an intermediate score, whereas parenteral nutrition has much less gene association and is therefore scored as highly specific. Finally, if you have a list of gene candidates or genomic regions you'd like to focus on, you can do that here as well. Now let's take a look at the main area of the analysis screen. Here, each line represents a variant, and all its associated information is summarized along the columns with interactive features. Normally, variant annotations would populate more than 100 columns. What we've done here is to classify all this information into the following categories: annotations and notes, genomic and genetic data, ACMG classification, variant calling quality and reliability, evidence, in-house annotations and variant allele frequency, effect and predicted impact on the protein, and frequency in control populations. Key annotations have been selected to be displayed to provide a concise single view, making the analysis much easier to manage. Other annotations are available upon expansion of each category. For example, under the genetic category, we can see also the HGVS, RefSec accessions, the codon changes, and dbSNP info. Let's look at the top variant as an example. You'll note that after we filter based on ACMG scoring and quality, it is the most likely candidate according to its phenotype score. The variant is located in chromosome 5, and clicking on its location will open the UCSC genome browser for this position. If the BAM file is available for the sample, you can also right-click to open up IGV and look at the reads in this region. The variant is located in the TTC37 gene. Since it's an OMIME gene with a disease, the gene name is bolded, and in brackets above it, we can see that the inheritance model from OMIME for this gene is autosomal recessive. Clicking on the gene will provide you comprehensive information about the gene diseases and other clinical variants in it and relevant links. If a gene has paralogs or pseudogenes in the human genome, a small P icon will appear next to the gene symbol. If the gene has imprinting information, the icon I will appear next to it. Clicking on these icons will bring you to relevant additional information. You can see the amino acid change is L761P, and clicking on it will bring you to EBI's VAR site, where you can find detailed information about this amino acid change and its impact. Under the ACMG group, you can see the classification of the variant for dominant and recessive inheritance models separately. Clicking on either will provide you with detailed assignment with evidence for each category, and the full evidence for the variant will appear on the right side of this dialog for your review. Under the Quality and Reliability group, we can see that the variant is a clear homozygous variant with a high quality and reliability score. GQ or QUAL scores are also available for expansion here. In the Evidence tab, we provide a phenotype scoring for each variant based on the strength of the association of the gene and the phenotype keywords entered for the case. This is also the default sorting for variants in the case of rare disorder diagnosis. Importantly, this score is accompanied by detailed supporting evidence from the comprehensive gene cards knowledge base, which are crucial for the variant review process. In this case, a disease called THE syndrome 1 is clearly associated with the phenotypes of the patient. You can see that phenotype keywords entered are found in the disease summary, in associated publications, and in HPO phenotypes. You can also find the number of matching phenotypes, summary of ClinVar pathogenicity, with dedicated links for more information, and the number of publications associated with this variant based on the Mastermind database. In-house annotations will display the number of times this specific variant has been annotated by your own lab in Geneyx Analysis. Clicking on this number will show annotations from other cases, which helps leverage previous work for better and faster analysis of new cases. The next column shows the number of times this specific gene has been annotated by your own lab in Geneyx Analysis, which can also be clicked on for more information. Finally, each variant will present the frequency of the variant and other samples from your lab in the system under the AF column. Next, we have the Effect and Prediction column, where you can find the predicted effect of the variant on the protein and the calculated severity of that effect. Finally, the Frequency group includes allele frequency annotations from a variety of public databases, including GNOMAD, ESP, 1K Genome, and Converge. In most cases, the most relevant variant based on the association to the case's phenotypes will be within the top few variants in this list, and in many cases, it will be the first one. In this case, we immediately found the causal variant in TTC37, which is associated with THE syndrome. Clicking on the gene, we can see that there are several other pathogenic ClinVar variants already annotated for this gene. Clearly, this TTC37 variant is a strong candidate. We can now set its relevance level and also add a note to the report about why this variant seems like a strong candidate. We can select the evidence for this to be presented to the clinician by simply clicking on the relevant information from the GeneCards knowledge base. Once this review is done, clicking on Report Preview will generate a visual summary of all the findings in addition to general information about the case. This is a great feature that enables the analyst to review the current results at any point and allows them to automatically generate a report with a click of the button. Importantly, reports are fully customizable and can include any information you need about the variants, the gene, the disease, inheritance information, and more. Geneyx Analysis provides template-based customization of reports to match your needs in formats that include PDF, Word, HTML, and Excel. When clicking on the Generate Report button, Geneyx Analysis will automatically generate two files: the full report in a PDF format with case information, candidate mutations with supporting evidence and relevant publications, and an Excel file summarizing the full variant tables that were analyzed. In this demonstration, we demonstrated how Geneyx Analysis can be used to rapidly identify a causal mutation in a rare disorder in a whole exome single sample with over 30,000 variants. Thank you for watching this demonstration. Sign up now and start analyzing your own NGS data at analysis.geneyx.com. Feel free to contact us at sales@geneyx.com.